Hello, today we're going to talk about a journal called Microbial Succession of Submerged Bones in an Aquatic Habitat. This is from the Journal of Forensic Science. First, let's go over some of the terms that I think are necessary for the public to understand. So we are going to be talking about a microbiome. What is a microbiome? Microbiome is the composition of all the microbial communities that are found within a particular environment. This could be bacteria, and it could also be fungi, which from here on we shall call mycobiome. The PMI, postmortem interval. The PMI is the time that goes by between the time of death and the time it is found. And it is very important in forensic science to be able to understand or reconstruct a death scene. Traditionally, these PMI estimations have been carried out using what's called a forensic taphonomy, which is the changes to the human remains, and also entomology by using insects. Ecological succession. Ecological succession is the process by which the structure of a biological community changes over time. Usually, when biologists or ecologists talk about ecological succession, they are referring to plants. So there have been studies in Mount St. Helens after the eruptions in the 80s to see how the bare soil was recovered by vegetation over time, started with lichens, mosses, and then greater plants, eventually trees. That's ecological succession. But microbial succession, well, in this case, will refer to how these microbial or these microbes change over time. What's going to be the composition of these microbes over time? Is it always going to be the same? Most likely, there's going to be different types that are going to appear at different stages. At the top, what we see on the images is actually um, entomological succession, insects that appear at different stages of decomposition, which have been a tool used in forensic science. It is important to know something about the admissibility of evidence in court in the USA. Microbial succession is not yet admitted in court because there is not enough information yet. But it could be used in the future, and it seems like it will be. Forensic labs are subject to International Organization for Standardization, in this case ISO 17025 rules. The scrutiny has increased because over time there's been wrong convictions, unfortunately evidence that is not conclusive or sometimes has been fabricated. The technique used here, or at least to read the DNA, is called the high throughput sequencing, HTS. So since 1977, Frederick Sanger is the first person to develop a DNA sequencing method called the chain termination method. After that, there were several advances in DNA analysis, but it will take a long time for this to take place. And because of that, it will also cost quite a bit of money. When the high throughput sequencing was invented, there will be great amounts of data that could be read. For example, they say here over 100 gigabytes of reads in 24 hours, also lowering the cost and therefore allowing for greater DNA analysis, much faster, much cheaper. Some of the research topic gaps that are found. So in the study of decomposition underwater, and actually most studies that exist on decomposition are focused on the pre-skeletal stages. So this means before it's just the bones. Let's remember this paper is about succession when there's bones in the bones because they want to look at long term, something that's been under the water for quite a while. So most studies have been about the previous stages where there's still some flesh, some organs in there. And also most of them have focused on terrestrial ecosystems. So because of that, more studies are going to be required before uh, microbial succession underwater can be used as a reliable method of evidence. It must be noted that the decomposition in aquatic ecosystems is different to what can be observed in terrestrial ecosystems. For example, the pH is going to be different. The temperature doesn't vary as much. The salinity is going to be different. The turbidity of the water, different microbial communities, and other vertebrates and invertebrate scavengers. So all that is going to change. If you go to different ponds, sometimes you may even find a different composition of organisms. All that needs to be accounted for. Also, the few that were found are limited to short-term studies, so studies that are from less than a year, and it's usually the body parts of Suscrofa, which is a pig or a boar. Only one long-term study was found by the researchers. So by long-term, they mean longer than a year in the topic that they were investigating, which is succession of microbial communities in bones under the water. But it was also incomplete because they only submerged a few bones and not the entire carcass, meaning they didn't wait for the entire carcass to decompose underwater, and that may just have a different effect because possibly certain microbes will come into the body before uh, the decomposition or before we reach the stage of just bones. So what is the relevance of this study? Why is it important? So here we have a piece of data from the World Health Organization that says that an estimated 236,000 people drown around the world every year. It's also one of the leading causes of death for young people between the ages of 5 and 24. Also, as we sometimes can see in movies or shows, but it happens in real life, that bodies of water have been used to dispose of human remains after a homicide. Or sometimes, unfortunately, some people choose them as a location to commit suicide. So this study can provide a valuable tool to understand what happened in this case of in these types of cases in the future. So questions leading to further investigation, to further research. How do different environments impact carcass decomposition? Can signatures of microbial invasion into bone be identified over longer-term decomposition? How do different components of microbial communities change over the post-mortem submersion interval? So these are questions that could be answered or could be addressed if somebody wanted to investigate a little bit more on this topic. This is their hypothesis. So predictable microbial community succession would emerge based on previous succession with aquatic composition in shorter terms PMSIs as well as with bones. Microbial communities within bones would differ from bone surface communities due to different environmental factors such as bioerosion and sources of exogenous taxa from the surrounding environment not immediately present inside bones. However, over the longer term, these communities would converge to similar community structure. This was their procedure, so they took 5 pig carcasses, so again, Scrofa domesticus, which they were used as human remains surrogates. So they weren't going to use people, they used pigs. They were placed under freshwater aquatic habitats to characterize the succession of the microbial communities as decomposition took place. They were placed 3 meters apart and at 1 meter depth. The sunken bones, which were left there after the whole carcass decomposed, so they remember they're focusing on the bones. They were sampled every 2 months over nearly a 2-year period. So targeted gene amplicon high throughput sequencing was used to describe the microbiome, which is the bacteria, and mycobiome, the fungi. Some questions for them to be answered or to be answered from the study were: how did microbial communities change and vary over time? Did the bone internal communities shift to an exogenous community structure over time that more resembled external microbial communities? How accurate are PMSI estimation models for a longer-term decomposition using bacteria and fungi? Finally, a little bit more about my opinion, what I think. So I think this methodology can be very valuable in the future for crime investigations because bodies that are found in the water can be dated more effectively this way. And I think that even though it's not admissible in court right now, if more people focus on these studies, eventually it may be that we will hear about it as being used as evidence in court. I do think that expert testimony will be necessary because let's remember members of the jury are members of the community and not everybody understands enough about the topic, and there will have to be a breakdown for them to understand what it is, what kind of evidence is being used here in the case.